All right, next we have our first Antonio, first but not last Antonio. We have Nino Chiocca. Maybe I'll go forward with the introduction while you set up. He has served as the neurosurgeon-in-chief and chairman of the Department of Neurosurgery at Brigham and Women's, and as the Harvey Cushing Professor of Neurosurgery at Harvard Medical School since 2012. Prior to these roles, Dr. Chiocca held assistant professor and associate professor faculty positions at MGH, and in 2004 became the first chairman of the newly developed Department of Neurological Surgery at Ohio State. He's a graduate of the University of Texas Medical School and completed his neurosurgery residency training at MGH. But I would be amiss to say, to not say that he is European and grew up in Padua. And with that, we're very much looking forward to your talk.
Thank you, Francisca. Great introduction. I appreciate it. Thank you, Forrest, for starting this great symposium. I love the fact that you have a systems biology group here on glioblastoma, this awful disease. So I'm going to tell you the story of a true academic bench to bedside approach for a particular compound. These are my, this is a glioblastoma clinical timeline. So Forrest gave us a great introduction to glioblastoma. I have the date there, 2025. That has not changed in several years. This is what happens fairly routinely. And I know the neurosurgeons in the audience know this, so please forgive me. But this is one of my patients, mid-40s, CPA, who had an episode of speech arrest, a father of two beautiful young girls. I saw him in the office, and he has this mass in his, as you can see here, in his left temporal lobe, left frontal lobe, close to the speech area. So what's usually done in the United States, in fact, in most countries of the world, is to take this out by surgery. So we take him to surgery, take this out, and then he undergoes chemoradiation, and then gets treated with adjuvant temozolomide. You can also give Optune, which is another device that one can give, and it's FDA approved. But unfortunately, this is very typical. Within 6 to 9 months, the tumor recurs. Most of these tumors recur locally. They don't metastasize. They usually recur in the surgical bed, like in this case, around here. And so what do you do now? Now this is when patients can undergo more surgery, potentially they can undergo more radiation, they can do a clinical trial. In this case, we took him back to surgery because he was still performing really well, and took this out again, and then started a clinical trial, but unfortunately this tumor just recurred within a few months. And you can see now at this point it's become unresectable. It's across the corpus callosum. As Forrest shows, the tumor is invading. It's basically going throughout the brain. And unfortunately passed away within a few months from this. So he lived what's the median survival for these tumors, which is about 15, 16, 17 months. So the median survival time for initial diagnosis is reported to be 15 to 20 months. This really varies based on genetic and demographic markers. For example, if you have a methylation of the MGMT promoter, you can actually, the median survival is up to 2 years. If you're older, you live less. If you're younger, you live longer. But we really have not made much of progress in terms of median survival. We know a lot about these tumors. These were one of the first tumors to be sequenced through the TCGA approach. So we have a lot of targets. But the clinical trials of mono-targeted therapies or even double-targeted therapies have really failed. There was a lot of hope for anti-angiogenic therapies. Those have failed. And even the more recent clinical trials of immune therapy that have been so successful, but a lot of the solid cancers, especially the immune checkpoint inhibitors, failed with this tumor. So that is what I wanted to discuss, is the failure of these immunotherapies. So very simplistically, one of the problems has been that these tumors are the prototypically immunologically cold tumors. That means that they're scarce of lymphocytes. You see very few T cells or tumor-attacking cells within the tumor, and these tumors are full of myeloid-derived suppressor cells and immune evasion particles. And so even when you're trying to make a T cell less exhausted, the T cell is just not getting to the tumor to attack the tumor itself. And so we've approached this by trying to change the microenvironment of these tumors. And so there have been several clinical trials that we and others have done trying to change the microenvironment of these tumors from a microenvironment that's full of myeloid suppressor myeloid cells and very few reactive T cells by intralesionally injecting either oncolytic viruses or gene therapies that express potent cytokines like IL-12 and others. Or some people have also now starting to put some anti-tumor gene proteins on scaffolds that will just stay within the tumor itself. And with this, we're hoping to change this microenvironment to one that's less immunosuppressive myeloid cells and lots of immunoreactive T cells. So for the people in the audience, if you want to go to sleep, this is my talk right here. This is what we're trying to do. You can go to sleep now. The compound I'm going to tell you about, we started working on in my lab over 20 years ago, and it's based on an oncolytic virus. This is a tumor-selective virus, and the original mechanism of action of these oncolytic viruses was thought to be due just to direct cytotoxicity. The virus infects a tumor cell, it kills it, and then propagates this infection, biodistributes, and kills the entire tumor. That's what we thought we were doing. But it turns out that because this is a pathogen, there's also innate inflammatory responses that occur. And so you have macrophages that get stimulated, lots of cytokines, lots of interferons, and a lot of these inflammatory signals have anti-tumor effects. In fact, in some cases, they even have anti-angiogenic effects. And so now we know that a lot of the mechanism, more than cytotoxicity, is this activation of innate inflammatory responses. But even recently, there's this thought that as you get this inflammation going into this immunosuppressive tumor, this tumor now becomes much more propitious for infiltration of T cells, but also recognition not just of viral antigens, but also tumor antigens. And through this process of epitope spreading, which I think is the holy grail for all of immunotherapy. Every immunotherapy you see up there, peptide vaccines, viruses, gene therapy, or CAR T cells, is counting on epitope spreading to get more T cells educated against other tumor antigens. So we're hoping to get an adaptive response against tumor antigens within the tumor. So this is the oncolytic virus we designed in our lab. So this slide shows you about 25 years worth of work. This is based on herpes simplex virus type 1, a common pathogen that's present in the human population. And there have been several herpes simplex virus type 1 oncolytics. One has been approved for melanoma. A different one has been approved for glioblastoma in Japan. We designed this, and it's a little different from the ones that have been approved. I'm not going to go into the specifics, but basically we're expressing one of the viral genes called ICP34.5 that's responsible for inflammation. This really does a lot of inflammation. But instead of having this inflammation occur throughout the brain, because herpes is neurotropic, we restricted this inflammatory insult to the tumor because we put under control of a promoter for the intermediate filament called nestin, which is highly expressed in gliomas, glioma stem cells, but the adult human brain is not expressed. So now we have this inflammatory gene, basically, that's expressed primarily in the glioma. So we want to cause a gliomitis, but not expressed in neurons in the normal brain, so we're not causing an encephalitis. And this virus can be turned off with antiherpetics. So we did from lab, got IND-enabling studies, got an IND with the FDA, all done academically, and then started this phase 1 clinical trial in patients with recurrent high-grade gliomas. Not just GBM, we also had some patients who had IDH mutant gliomas that were grade 4, as well as some recurrent anaplastic oligomas. But the majority were GBMs, glioblastomas. So we first did what's called a dose escalation trial, where we did a single injection. We're not giving this IV, we're giving this by direct intratumoral injection of this virus called, we can now call it CAN310. What you'll see in these slides, I'll call it herpes, oncolytic, CAN310, sometimes it says arcunescent. They're all synonymous, they're all the same thing, okay? And so we went from a dose of 10 to the 6th, 1 million viral particles, all the way 10 to the 10th. We did 30 patients. We did not see toxicity. Actually, this was really well tolerated. Most patients went home the next day. I actually had some patients fly in and fly out. So really well tolerated. And we got all the way up to 10 to the 10th plaque forming units. After this, we dose expanded. We treated another 12 patients at a dose of 10 to the 9th, a little bit less than 10 to the 10th, just because I did not have enough vials to do additional patients at this high dose. And we finished this phase in 2021. This was published in 2023, this clinical trial, and that's actually what I will discuss, the results of this trial that was published. And this we called Arm A. We had a second arm called Arm B, where we actually pre-administer cyclophosphamide to immunomodulate the patient. I will really not discuss that, and this paper is in preparation. And then through this consortium called Breakthrough Cancer, Michael Chima, Forrest participated in this. They've really provided us the funding to do a serial injection and serial biopsies over 4 months for an additional 12 patients. So in these 12 patients, we're doing time-dependent injections, and at the same time get several serial biopsies so we can see what is happening in these tumors as we treat the patients. And so far, we've accrued 8 out of 12 patients for this. And the analyses of these, we have over 400 biopsies. The analysis of these biopsies, serial biopsy, are undergoing right now. So I'm going to show you a video because I told you patients tolerate this really well. I'm going to show you a video of a patient. She's going to tell you her story with this trial. In the middle, it breaks and goes to another patient, and it's actually her daughter that's speaking, because that patient, who was her mom, did extremely well, but unfortunately passed away 2 years after treatment in a car wreck. She was actually not the driver, or the husband was the driver, but her daughter will tell you the story about her mother's experience. This video was shown on a local TV channel, and I know they named me, so it's my 5 minutes of fame, but really what's important is the patient and the patients. I don't know what happened to the audio. The audio was working before. No. We tested before and the audio worked. Is that the problem? Maybe shut off. Yeah, try it. Let's see if that works. No. All right, we can just skip it. I'm just going to skip it. Okay, so this was really well tolerated. First 42 patients in that arm, 30 plus 12, did no dose-limiting toxicities. This was a phase 1, so it was not designed to look at safety. But like every phase 1 trial, we were looking for any tidbit of information that we could get from these patients to see if there was anything, any demographic marker, any cellular marker, any genetic marker that could tell you which patients did best. So one of the things we were collecting prospectively were antibodies to HSV1. As I said, this is an HSV1-based engineered virus. And patients, but in the human population, there's about 2/3 of us have antibodies to HSV1. So interestingly, when you look at the seropositivity of patients, we're actually surprised to see that the patients that were seropositive, and that's the curve in blue, were the ones that did best. The median overall survival time was over 14 months. for recurrent GBM that's not resected, in other words, we were just injecting these GBMs, the median overall survival time is more in the 7 to 8 month range. Okay, so we kind of doubled that. And when you compare to the HSV seronegative patients, the patients that did not have antibodies, their median overall survival time was 7 months, which is exactly what the historical median survival time was. So we were surprised by that because the oncolytic herpes virus that's been approved for melanoma in the US and the one approved for GBM in Japan did not show this. Actually, if you had antibodies, you did a little bit worse, probably because people think that the antibodies got rid of the virus. But it was not statistically significant. So that was kind of an interesting finding. So then I said, well, maybe there's something different about these patients. Maybe these patients have something else. And so we did these parametric Cox proportional hazard ratios to see all the potential prognostic factors that could differentiate this population to this population. And even when we did this, the serology to HSV1 showed here was independently significant factor for survivorship compared between HSV1 positive and negative. Age was significant too. The size of the tumor was significant too, but all the others were not. So this is interesting. So serology, if you're seropositive, you did better. It seems to be an independent prognostic factor for that. The next question I asked, well, maybe there's something else. Maybe it's not this. Maybe it's something else. So luckily, we were also collecting antibodies to a related virus, HSV2, as a control. I don't know why we were doing that. I just had put it in our protocol. So we collected those antibodies. So we checked to see if seropositivity to HSV2 had anything to do with this. It did not. Actually, the patient with HSV2 seropositive probably did a little bit worse, but this was not statistically significant. And then at the same time, Steve Francis, who's an epidemiologist, genetic epidemiologist at UCSF, published a paper in Oncology where he had a large data set of gliomas and glioma patients and was looking at seropositivity to a number of different viruses. Now, these are just a general population. They're not doing this trial. They're doing something else. And they published this paper showing that HSV1 serology is not associated with GBM overall survivorship. And so this seems to be very, very specific to this particular compound, CAN310. So this is interesting. We'll have to prospectively validate that to show that this is really true in another trial. But the second thing that was most interesting to me was the fact that we really tried to get back tissue. So this is a sort of a timeline of what happens to a patient. So these are all recurrent GBM. Sometimes there's second, third, or fourth recurrence. So at time 0, we would inject the virus into these tumors. And then sometime in the future, these tumors seem to recur. They had more enhancement. Maybe the patient was doing clinically worse. So we really tried to get back tumor, either by surgery. In a few instances, the patients donated their brain, so we also had autopsy tissue. And we also could collect blood at all these time points. And then could do all of these complex multi-omic analyses to see whether there was anything that correlated with outcome. And so at the beginning of this, I told you that when we put a virus in here, we changed the microenvironment. Is that true? Most of what I told you was based on mice. Is that true in humans? So the first question we asked, is there evidence after injection that you see more CD8s, more CD4s, more B cells in these GBM infiltrates? So again, we were comparing before injection to sometime after injection. So you can do this visually. So this is a patient before treatment, patient 16 here, before treatment, and then I think she got resected like 4 months later. And what you can see is that you see all these CD8s and CD4s. Sometimes these are 3 different patients, 16, the same one as here, 19 and 21. So what you can see is that you see perivascular accumulation of lymphocytes, thinking of that these cells are coming to the bloodstream and then trying to expand into the tumor, infiltrating the tumor. Sometimes you see it in these perinecrotic areas. But because we had about 30 of these paired samples between before and after, we could also do some quantitative analyses. So these are quantitative analysis, these paired samples before and after. And you can see there was a statistically significant increase in CD8s after injection, in CD4 after injections. B cells, we were not statistically significantly increased. Some patients had lots of increases, some were much less so. You see here the averages are here, but again, there was a little bit too much of a range to make this significant. What is interesting, can you correlate that with anything? You see more CD8s, more CD4, you've injected a pathogen, great. So the next question we ask, is there any correlation between the infiltration of T cells and survivorship outcome? And so this is work by Alex Ling. Alex was a postdoc in my lab, and now he's a junior instructor at the Brigham. But what Alex did is started putting on the x-axis the change in CD8s in these specimens after oncolytic virus injection, or the change in CD4s, and then looked at survivorship as a continuous variable. So this survivorship is a continuous variable, which is a more, robust statistical test than just saying long-term survival versus short-term survivorship. So each one of these dots is a patient. And what you can see is if you look at all the patients that we looked at, it was not significant. But if you looked at the seropositive patients, the ones that lived the longest, there was a significant correlation between these changes in CD8s, the change in CD4s, and survivorship. So again, what I showed you so far is that seropositivity seems to be involved with survivorship. You have more T cells in there, and in the seropositive patients, the more T cells you have in there, the longer your survivorship. So I said, well, let's do another test here. Let's try something else. So we did what I call the $250,000 experiment. We made genomic DNA from our specimens, sent it to Adaptive Biotechnologies, which can then sequence from paraffin samples the T cell receptor, particularly the beta chain of the T cell receptor. So now you have what are called T cell clones. And we correlated the T cell clones with survivorship. And so here we're looking at the tumor TCR betas again. And we're looking at survivorship as this continuous variable versus a change in the tumor T cell fraction. This is just an indication of the change in tumors. And again, what we could see is that there was a statistically significant increase in survivorship the more of the T cell clones were in there. And this was also related to T cell diversity. Diversity is a measure of a metric of T cell diversity, which is how well the T cells respond to different antigens, for example. And the more diverse they were, the better the survivorship. And this was also seen in the PBMCs from the same patients. Again, there was a statistically significant increase in survivorship the more of these T cell clone types you saw coming into the tumor. And again, this is also associated with survivorship. So what I've shown you so far is that there's evidence of increased CD8s and CD4s. We've really changed the microenvironment of these tumors with more T cells. The more T cells that come in there, either just counted grossly by immunohistochemistry or looking by T cell clone types, the better the outcome. And the last thing we asked is there any changes in the tumor immune signatures by bulk RNA sequencing? Are these changes correlated with outcome? And so this is a complex slide, but basically, here we're looking at p-values. And so this red bar is a p-value of 0.05. So everything to the left is significant. On the y-axis, we're looking at 13 different immune signatures by transcriptomic analysis. And what I'm just trying to show you is that in the seropositive patients that we could study in blue here, all these signatures are positive after injection of the virus. So there's definitely a change, and most of these are immunoactivity signatures. In the seronegative patient, you don't see as many, as shown here in red. So the way we put all this together is that really what we're doing here is an intralesional booster vaccine. In other words, in patients that have been previously exposed to herpes, that have antibodies and probably T cells that recognize the virus, maybe in a central memory pool. When we inject the virus into the tumor, you start getting a profound pretty rapid antiviral immune response. Maybe it's antibody-mediated, maybe it's T-cell mediated. I think it's T-cell mediated personally, which starts clearing virally infected cells, particularly tumor cells. But we also think by epitope spreading also some tumor bystander cells. And clearly you can get a better response in that aspect. However, if you're seronegative and you inject the virus, these tumors are growing fast. There's just no way for this viral therapy to really catch up. So these are the conclusions. We can change this tumor microenvironment with injection of this virus. We actually see these inflammatory infiltrates even several months. We've seen as late as 3 years out. So we can really turn these tumors into some durable cold to hot tumors. We have now treated a total of, I think it's 61 patients, and we have not seen neurotoxicity. We can see that survival patients treated with this oncolytic associates with positive HSV-1 serology, although we do have to prospectively validate this. These patients are the ones that also have more increases in T cells and T cell clonotypes. We actually have some specific TCRs that seem to be associated with survivorship. We see more immune signatures. And I didn't show you this, but the seropositive patients also clear the virus. We do not find virus in the seropositive patients, even the ones that live long. But in the seronegative patients, we find virus even several months to years later. We're doing additional analysis, single-cell RNA sequencing codex with the SUVA lab, the Tirosh lab, and the Wuka Finning lab. And we're now in the midst of doing this longitudinal sampling and injection of multi-institutional trial, which is funded by Breakthrough Cancer. Do I have time? So I'm just going to tell you, so these are all the individuals. Again, it takes, I don't say it takes a village, it takes a city. Over 20 years, they've worked on this. A lot of the funding has come through the NIH, ACGT, philanthropy, and there's multiple individuals that have been involved in this. Now, I just wanted to really give a shout out to Breakthrough Cancer. This is a collaboration between MIT, Dana-Farber Brigham, Memorial Sloan Kettering, Johns Hopkins, MD Anderson, where we're doing this longitudinal sampling platform to try to understand what is happening in the tumor microenvironment as we do a therapy. Initially, we wanted to do a systemic therapy, but we thought that putting patients through 6 or 4 multiple injections without treatment may not be approved by the IRB. So we added this intratumoral injection as a therapeutic so we could also do these analyses. And so the idea here, again, is that when you have a therapeutic, you could do these serial time point injection assays in these tumors and then analyze this by multi-omic approaches to see what is happening in the tumor itself. You can also collect CSF. You can also collect blood to see if you can associate the biofluid changes with what you find in tissue. So actually, we've done 8 patients. Patient number 7 and 8 are currently undergoing treatment. But these are the first 6 patients in the first cohort that have undergone this. And this is definitely feasible. In some cases, like this patient, we got up to 26 biopsies from 4 different locations over 120 days. In some cases, we stopped early because either there was a little bit too much, looked like tumor progression, and so these patients either underwent surgery to resect the tumor, or, and in fact, a lot of times we actually found a lot of inflammatory pseudoprogression, more inflammation than tumor, or they decided to do something else. And one of the questions, what can you do with some serial samples? I was talking to Antonio before, and actually you can do a lot. You can do lymphocyte immune analysis, you can do proteome analysis, you can do RNA analysis, you can do DNA analysis. And this really involves a gigantic team, over 100 scientists over these 5 institutions that are doing these studies. And some of them are shown here in this slide. Each one of these cores goes to a bunch of different labs, including the ones at MIT and Dana-Farber, but also MD Anderson, Memorial, and Johns Hopkins. So patients, we have more patients than slots on trial. That's my major, as the IND sponsor, that's my biggest problem, is always trying to find a spot for patients that want to go this. We have not had serious adverse events or dose-limiting toxicities. We can get multiple sites from tumors. We've been able to get each site from 6 to 26 biopsies. We have, right now, with 8 patients, we have over 400. And each one of these is of sufficient quality and quantity to get great scientific data via omics approaches. So thank you, and thank to the entire Breakthrough Cancer Labs. Some of them are here in the audience. I appreciate it.
Fantastic. Thank you so much. Questions?
So, Nino, you mentioned this briefly, but maybe you could go over it a little bit. The CAN-3110 immune response seems different than other herpes virus therapies that you are that you compared to. So what is it again that's different?
What's different is the genetics of the virus, how the virus is constructed. So our virus, it still retains some of the immunoevasive properties. It still has this one gene called ICP47 that allows MHC to downregulate. So actually in the infected cells, it's pretty immunoevasive. That allows it to be more robustly replicating, number one. And I think that that's why you get this importance with the serology. The other ones are pretty feeble. They don't have that, so they get recognized pretty fast, I think, by innate inflammation and get removed by the circulation. In fact, with the TVEC, which is a melanoma trial, they can never find virus if they ever reset these tumors, whether it's seropositive or seronegative. So I don't think these viruses hang around very long.
So precisely the thing that might have scared you from a safety point of view.
Yes.
Actually
and that's why the FDA made me start at 10 to the 6th by alpha log, using an intraoperative MRI to make sure that the injections were in the right spot. But it's actually been extremely safe. I mean, some of the other immunotherapy trials, patients are really sick. They're in the hospital for like several weeks. I'm not going to name the immunotherapy trials, but you can imagine. With this one, I've had patients fly in across the country, get their injection, and fly back the next day. So,
Nino, I imagine you didn't do any pre-selection of the patients initially, and I was wondering if you had any consideration of the status of the tumor before treatment to understand the response. In other words, also, you seem to allude mostly in your talk to the state, the cold state, right, of these tumors that then become immune hot with more infiltration of immunoactive cells. But as you also mentioned many times, some GBM especially can have really a large infiltration already in the beginning diagnosis of immunosuppressive myeloid cells. What happens to those cells? Have you checked if there is a role of the myeloid cells in predicting the response?
Yeah, so in the first study, this RMA study that I showed you that was published, the only way we could really look at myeloid cells well, because we tried to do it by immunohistochemistry, but these stains are crazy and it's very hard to enumerate. They're very complicated, and I gave up. But you can do it by transcriptomes, So we can clearly see, particularly in the seropositive patients, that there were many more, if you believe the M1 to M2, M1-like transcriptomic signatures after injection of the virus. So I think there's still myeloid cells in there. They're just much more immunoactive. These is a mixed bag of gliomas. They were recurrent. Some of them had previous treatment. But again, all that I've shown you is the GBM. They're IDH wild type. GBMs are not the others. Yeah.
Thanks. Hey, Nino, I'm sure you're here because one of the next steps is to somehow combine this with, the previous talk, right? To look at, are you actually targeting more migratory cells or more proliferative cells with this, and what's with the signaling pathway? Because you actually have shown that there is some extravasation and some, higher density of whatever you do around the blood vessels, which makes a lot of sense. So that's a comment. And then the question is, I'm super surprised that there's no edema like, 25 years ago. And, is this because all these patients are you know, it's a recurrent tumor and there's some cavity and, you know, there's not a lot of pressure that, you know, is being generated? Or what do you see on MRI?
No. So the virus, there's edema. The virus does cause edema.
But there's no headache or there's no headache or no seizure activity?
So there were no DLTs. The worst SAEs, serious adverse events, were persistent seizures in 2 patients who were young and were IDH mutant gliomas. They were not actually GBMs. That's been the worst. We've had some headaches. We've had some, you know, so we saw patients even a few months later that showed more enhancement. So we took them back to surgery. When we take them back to surgery, there's a lot of inflammation. There's a lot of lymphocytes. It's mostly tumor confined. But there is edema. There isn't, I mean, this is an inflammatory agent. It causes edema around the tumor. Yeah, so the way we inject this is right into the gadolinium-enhancing area. Sometimes maybe we get flare. I think the virus is just providing an initial, how do I say it? It's the initial match that starts a process. The virus is not going, well, I shouldn't say this. In theory, the virus should not be going away from that. We do have a patient that was actually in the paper who had multicentric glioma. We injected 1 tumor, and that tumor became necrotic on MRI scans over time, but he had a second tumor temporal lobe that kept on growing. He donated his brain 8 months later, and when we did the autopsy of his brain, the tumor that was going at you was full of virus and T cells. I have no idea how the virus got from the injection site to that site. There were 3 other spots that you can actually see on the MRI scan, but there was some tumor there in 3 other spots in the contralateral hemisphere. There was no virus by immunohistochemistry, but there was virus just in the tumor, not in the brain, by PCR. So I actually don't know how it got there. And there's a postdoc in my lab that's got all these ideas and working on it. He's been at it for 2 years now, so I'm not sure.
It's interesting that the 2 patients who had seizures were IDH1 mutant. Do you think there's any biological significance to that they would have more edema and therefore more seizures?
No, well, I think that there's some data, the IDH mutant patients, first they're younger, you seem to have more seizures. So I think they had seizures even before. The seizures that got worse afterwards. And in both cases, we actually gave the patient acyclovir because we thought it was because of the virus. Finally, we got the seizures under control.
Okay, excellent. We have 4 questions online. Given the success, Divya Senya, given the success of oncolytic virus in this case, curious to see if you have seen or would expect to see similar anti-tumor activity in other mimicking non-viral models of cytosolic delivery, such as mRNA peptide vaccines. Wondering what the principal mechanism that makes this work and if that would be leveraged for customizing a more effective immunotherapy.
Can you say that again?
Maybe easier to read it, actually.
That was a long question.
Yes.
Yeah, I don't know. I mean, I think that there's, so what they're asking is if you put a mRNA vaccine or a peptide vaccine that mimics, maybe mimics viruses, could you see a similar effect? I don't know. I think there may be some common mechanisms amongst all these inflammatory, whether it's a cytokine, a virus, or maybe even a CAR T cell. And then there's maybe different mechanisms. I think the problem with the mRNAs and single peptide vaccines is that they're very targeted. This is pretty untargeted. This is just, you start setting up an inflammatory response in the tumor, and then you see what the immune system does. So I think those will be the differences.
Excellent. And then James Vega-Lucarelli, are there more tumor-specific CD8 T cells in the virus-treated group? And did you look at the impact of treatment on glioblastoma stem cells?
So there are more. Yeah, definitely. That's we saw just a tremendous influx of CD8s and CD4s correlated with outcome. You can also do this by genomics. What was the second question?
T cells and stem cells.
Yeah, that's difficult. With glioblastomas, the stem cell hypothesis is that there are some stem cells in there that you can look by nestin expression. Again, this virus is driven by nestin promoters, so in theory, it should definitely infect glioma stem cells. But in humans, it's very hard to do that and figure out whether the stem cells are getting infected, just grossly looking at that. And you almost have to like get them out of culture and put them in culture. In culture, if you believe in glioblastoma stem cells, the virus definitely infects them. But I don't know in vivo.
Excellent. Last question from Chelsea Cherigino. Great talk. Do you believe that there would be any correlation between SNPs in the tumor cells and length of survival in patients?
Yeah, we didn't look at that. I mean, there's some data about GWAS and SNPs potentially correlating with survivorship. I'm not sure if there's that solid, but we did not look at that. Thank you for your questions.